Now that we've got the refresher of pharmacogenetics under our belt, let's switch gears very briefly and talk about something that I personally study in my lab, epigenetics. So epigenetics has been around for a while. It first was coined by a person named Waddington in 1942. And what epigenetics is, it's basically how we use the same genetic code throughout our entire body, right? But somehow our body knows to use the same codes to make brain cells, to make nerve cells, to make red blood cells. So despite being the same exact genetic blueprint throughout the body, we can make these drastically different cells. And so how this happens is through epigenetics, meaning on top of the genetics. It's some sort of regulation occurring that's outside of the letters A, C, G, and T that we see in DNA. So it's a change in gene expression that does not involve a change in the underlying DNA sequence. There's really three major types of epigenetics that are out there. The first is DNA methylation, which we will talk about today. We won't talk about histone modifications, but they are involved in epigenetics. And essentially what they are is changes in those histones we talked about in the first lecture. It's changes in the tails, changes in molecules that are added or taken away, and that changes the way they function and control gene expression. And then additionally in lecture 1, if you remember, I talked about those three main types of RNA, and then I briefly kind of threw it away that there are a bunch of other RNAs. And so those a bunch of other RNAs are actually critical in controlling gene expression in various mechanisms. So, for example, there are things called miRNAs, which are these short RNA kind of sequences that go in there, and they can actually cause mRNA degradation and transcriptional repression. Additionally, there are other things called long non-coding RNAs, and these interact with histones to produce what's called chromatin remodeling, which can change the way that the chromatin makes genes available for expression. So again, if these are areas of interest to you, I suggest you go look them up. They're very interesting and intense and complicated. However, they're very active areas of research. But today, we're going to talk about DNA methylation. So what is DNA methylation? And it's the addition of a methyl group to the 5 position of cytosine. Okay? And that occurs by certain enzymes called DNMTs in passive DNA methylation. And you can actually have active DNA methylation as well through TET enzymes that join the process. But in general, DNMTs, there's certain subtypes that actually do it. They actually add that methyl group onto the 5 position. It's been called the 5th base because it actually adds more complexity to the genome and really has tight regulation on the genome. 80% of DNA methylation occurs at what are called CPG dinucleotides, so a C occurring next to a G. These mostly are where the DNA methylation occurs in the genome. It's distributed throughout the genome. 70% are actually in a methylated state. And so it can vary across the genome, but actually most of them are methylated. And again, there are the enzymes that are responsible for passive or active DNA methylation. Now there's an actual kind of geography to DNA methylation, and that geography is called CPG islands. And so what CPG islands are, they're very dense groups of CG nucleotides that may or may not be methylated. In general, they're found in promoter regions, and in general, they're frequently hypomethylated. So these dense group of CPG dinucleotides, they're usually hypomethylated. However, if they're found within gene bodies, they're generally methylated. So it really just depends on the gene you're looking at. It depends on the gene area you're looking at. So again, just like genetic variation occurs throughout the genome millions of times, this adds an additional layer of complexity where methylation marks occur millions of times throughout the genome. So again, another area of research is actually combining genetic variation study with DNA methylation to understand its effects on phenotype. I just want to note that there's alternative methylation possible. And so what I mean by this is they now have discovered, several years ago now, but they've discovered other forms of methylation: hydroxymethylcytosine, formylcytosine, carboxylcytosine, and they're all involved in kind of the DNA methylation cycle, if you want to call it that. And so each one, they used to think that the demethylation cycle just produced these kind of intermediates that had no function, but now they're finding that each of them seem to have their own regulatory function. So it's an active area of research. It's not going to be clinically applicable for many years, but it will be someday. I think they're going to be utilizing this for drug treatment targets and things like that. So finally, what is the effect of DNA methylation on gene transcription? And in general, when you have DNA methylation, it usually causes gene repression. So how does it actually cause that? When you have methyl groups on your DNA, it crowds the DNA. It blocks those transcription factors from coming in. It blocks the RNA polymerase from being able to dock. It can actually recruit these things called methyl binding proteins, which further block. So there's a lot of action that just these simple marks on cytosines can cause. So again, the really take-home point here is to remember, in general, methylation is associated with DNA repression. However, obviously, just like everything in science, there are exceptions to that rule, but in general, methylation, repression. Now that kind of concludes our talk about epigenetics. I really just wanted to get it in your head so that you kind of know it's there. So in 20 years, when we start to see it in clinical practice, you'll be like, yeah, I know what pharmacoepigenetics is, because Dr. B taught it to me 20 years ago. So now we're going to move to just giving you a quick overview. I'm going to log in and show you some of the main resources that I think will be very useful to you in this class. Okay, in terms of pharmacogenetic databases, again, I'm just going to go through a few of them and kind of give you an overview of them and really show you what I think are the most highly useful ones for you as you study in this course. So the first one and the best one out there is called PharmGKB. And so it's just pharmgkb.org. You can Google or do whatever search engine you use. If you do put them into the search engine, they'll show up. So PharmGKB, the first time you log in, they'll actually offer a tour for you to show you the homepage, but I'll do it for you. So it's just like any database. You can search whatever you want in there. It shows you kind of these highlights at the front where they've annotated 696 drugs with pharmacogenetic information. They have 149 curated pathways. They have clinical guideline annotations for 143 drugs and drug label annotations for 753 drugs. So there are a lot of drug information for pharmacogenetics out there. You can look at what is pharmacogenetics. You can look at what the actual PharmGKB is all about. And what I'll tell you is that it's actually a curated knowledge base by PhD level scientists where they continuously, every day, every week, I'm not sure how often they do it, they curate the literature and they add it to this database. Again, you can see all the annotations. They kind of repeat this down here, but they have a lot of good information here. And additionally, you can have different resources like specific disease pharmacogenetics. You can have gene-specific information tables, etc., etc. And the partners that work with this are CPIC, which we'll talk about next, the Pharmacogenetic Research Network, PharmVar, which is a different website you can use, and PharmGKB. So let's do an example here. Let's look up the drug clopidogrel. So as you type, things will come up. And it doesn't just have to be a drug that you search here. You can search a gene. You can search an RS number. So it's a very, very useful website. So let's click on clopidogrel and just give you an overview of a typical, this is a really high quality pharmacogenetic drug. So there's a lot of information here. It's not always like this. So they give you an overview. Its prescribing infos included 9 different prescribing infos. It has 5 different drug label annotations. It has 45 clinical annotations, 2 different pathways. Then it gives you the structure of the drug. It has a PharmGKB ID. Then it basically gives you an overview of the drug. What's the genetics, what's the PK/PD, and the PGX associated with this drug. It'll go through any molecular properties and metabolites as well. So you can click on these links, but we'll go down through the left side here. So going right to prescribing info, it always starts out with the biggest level of evidence, which would be clinical guidelines. And the reason I would call clinical guidelines the highest level of evidence is specifically because in order to get a guideline, you have to basically have the highest level of evidence. And we'll look at a level of evidence with PharmGKB right when we finish this part. So there's 3 different guidelines with clopidogrel. Again, this is not typical for all drugs. Clopidogrel is very well documented for pharmacogenetics, and we'll do a case on it as well. You have different annotations as well as you go down through the prescribing info, including FDA label annotations as well, which is on the next page. So drug label annotations, it tells you all the different bodies across the world that have pharmacogenetics in the clopidogrel drug label, all the way from the FDA, Europe, Japan, Canada, and the Swiss. Apparently they're not part of Europe. I'm not sure about that. And so it also gives you a PGX level, which is actionable, clinically actionable, suggested. There are different levels too that I will show you on the next page. Clinical annotations, that really refers to looking at the specific variants within whatever drug you're looking at and their association. So you can see they give levels, they give the variant, the gene of the variant, the molecule, which is clopidogrel, because that's what we searched here, the type of clinical annotation it is, and then the phenotype it's associated with. So again, these are really highly curated, really good information here. And then it'll associate any sort of variant annotation. So it basically lifts off the clinical annotations and then brings you to just look at the variant annotations. So this is useful when you know you need to look at certain star alleles or certain RS numbers, and you want to find more with a specific drug. So instead of going to PubMed, which is kind of difficult, if you were to put this in, you might get hundreds of hits, thousands of hits. You can come here to this curated information, and it'll actually tell you the associations, the p-values, the type of study it was. It'll actually give you a link to the study if you click read now. They actually put the case numbers, the allele numbers, etc., etc. So it's really awesome information that enables you to not read an entire paper, but to get the gist of it. On top of that, they have different literature on clopidogrel and pharmacogenetics. They'll show you pathways. This is pretty useful if you need to quickly understand the pathway of clopidogrel, and then the stars in there is where pharmacogenetic information is available. And then I have a related to button where it'll take you to related molecules and automated annotations. Here is the possible relationship between a variant and a drug in a sentence from an abstract in PubMed. So really this is just an aggregator, probably not as useful here. Okay, PharmGKB actually gives you clinical annotation levels of evidence, with 1 being the highest, meaning that there's some sort of guideline or known clinical implementation. Level 2 means it's a very important variant. It generally means that the evidence associated with it is quite good. There's probably some replication there, but it's not quite perfect enough to meet guideline standards. Then you have level 3s and 4s, which is there's some sort of association that's put out there in the literature. Either it's been negated in other studies, or the studies with the associations are low quality, meaning cross-sectional or retrospective. So the next website I want to show you is called CPIC, or Clinical Pharmacogenetics Implementation Consortium. It's cpicpgx.org. And CPIC is great because you can also find CPIC guidelines in PharmGKB, which we were just in, but it's really good just for coming straight to the CPIC website, clicking on guidelines, and then you can search for your drug or your gene, or you can scroll down. You can see they have about 24 guidelines available. So this is a great thing to know. And all CPIC guidelines have the same exact layout. So you click on it, it'll tell you the guideline. This one's for SSRIs 2D6 and 2C19. And then you can either click on the open access paper, and all the guidelines are the same. They go through the background on the drug, and then they tell you how they assigned genotypes and phenotypes. And then they actually give you a table that gives you dosing recommendations based on the gene and the drug. So all CPIC guidelines are the same. They're all going to have this table that essentially tells you what to do based on a given genotype. Other areas of importance on the CPIC guideline page is their supplementary tables, which are really useful. For example, they have what's called translation tables. So how do you figure out what makes up CYP2D6 star 5, star 9, star 41? This is this diplotype translation table and diplotype phenotype table. That'll tell you how to do it. So just remember that. That's going to be important for you guys. And it also usually has some sort of race and ethnicity table. I believe that's usually S2. Let's see. Oh, no, S3. And so that'll tell you the frequency of particular variants depending on the race and ethnic makeup of a given population. One thing I did want to mention after we talked about CPIC is that PharmGKB does this really cool thing with any CPIC guideline where you can click under prescribing info for a given drug. So we're in clopidogrel. You click read now on that CPIC guideline and you can actually pick the alleles that you want it to tell you the CPIC recommendation for. And it will tell you. It'll say recommendation alternative antiplatelet therapy. So this is a really easy way, I guess if you don't want to go look up the CPIC table, to find the CPIC guideline recommendations. One other thing I wanted to mention was how CPIC assigns its actual guidelines, how it prioritizes them and begins to make them. So it does a combination of that PharmGKB levels of evidence I just showed you. And then it also does that FDA-approved drug labels, which we saw in PharmGKB, which is actionable pharmacogenetics, genetic testing recommended, genetic testing required, or non-actionable. So CPIC looks at this, and that's how they prioritize their future guidelines to come out. Another really useful website for this class is called Snipedia. Sometimes I call it Snipipedia, but I think it's Snipedia. Essentially, it's a really intuitive, just like Wikipedia, kind of aggregated website, but it's nice for certain things. So, for example, if you need to figure out what star alleles are composed, what SNPs are composed of CYP2C19 star alleles, and you can't find the translation table in PharmGKB, you could try to look it up in Snipipedia. So let's just try to look up 2C19. So you can see here, it gives you an overview of 2C19. It tells you certain things over here, like the RS numbers that are involved. And then what's nice about some of the information in Snipipedia is that it'll tell you what RS numbers make up what star alleles in here. So you can see star 2 is made up of this RS number versus star 3 could be this one or this one. Star 5 is this one, and so on and so forth. So a lot of good information that can be found in Snipipedia. It's pretty good for what I would call non-standard drugs, so maybe OTC or dietary compounds. It can be really useful for that. Another website that's pretty good for searching reference SNP or RS numbers is dbSNP. You can actually just input an RS number here, hit enter, and then click on it, and it will tell you important things that you might be interested in, such as what are the alleles that make it up, what is the minor allele frequencies, things like that. And they have side tabs too that you can click on, but this is much more experimental, probably a little less clinically applicable. There's a database out there that's really an aggregator, I would call it, and it's the drug gene interaction database. So you can actually search drugs and genes. So let's look up CYP2D6, paroxetine, and you can limit it to different types of filters. But let's just hit find. What it does is it pulls down different resources here based on what your input was, and then you can click on it and go through it like that. So this might be somewhat useful if you need to just trying to figure out where to start, or you're trying to look up a new drug and you're trying to see any sort of genes that are with it. But you can do this essentially the same thing in PharmGKB, which is reflected in the fact that half the results here are PharmGKB. ClinicalGenome.org or ClinGen is essentially just a curated NIH-funded database that looks at any sort of genetic variant and its association with any sort of disease or drug. So it could be useful for some sort of pharmacogenetic information, but this one's usually a little more useful for just disease states associated with genetic variations. And then finally, if you just search genome glossaries, There's several out there, and they're mostly supported by the NIH, but it can be pretty useful for different terms that you might run into that you don't understand or that are new in papers that you'll read. So just keep this in mind, that this might be a useful area to come if you run into genetic terms in your paper reading that you don't understand. Okay, well, that wraps up our overview of pharmacogenomic resources. Again, I highlighted the main ones that I think will be useful for you. There are other ones here that you can look up, and you probably can find many more if you just search the internet. Okay, thank you.